Hello, my name is Marco Cavaliere, and this afternoon I'm presenting a research about the environmental monitoring of the Gulf of Bagnoli using benthic foraminiferal metabarcoding and morphology-based assessment. Benthic foraminifera are single-cell organisms that are commonly used in many fields of earth and environmental sciences. In particular, they can be used as bioindicators because they react very quickly to environmental changes. Bagnoli area is a large brownfield close to the city of Naples, southern Italy. Bagnoli hosted steel, concrete and lettering factories which operated till 1992. Since then, many remediation projects were proposed to restore the area. Previous studies found high concentration of heavy metals, for instance mercury, lead and zinc, in the sea sediments in front of the former plant. We collected 12 sea sediment samples using a box scorer at different depths, from 10 to 40 meters. The abiotic analysis performed were the grain size analysis, the geochemical analysis to reveal the heavy metal concentrations, and the organic matter characterizations. While the benthic foraminiferal analysis were based on a double approach, firstly the eDNA metabarcoding, and secondly the classic morphological based analysis. The Spearman's metric of correlation clearly highlights significant correlation between abiotic indexes, here we have a pollution load index and the total organic carbon, and many ecological indexes. In particular, the foramambi and the genetic foramambi, G foramambi, are positively correlated to the pollution load index and the TOC, as well as the TSI, the Tolerant Species Index. On the other hand, there is a negative correlation between abiotic indexes and the foram stress index and the molecular modified Shannon-Weaver index. The environmental stress can be considered as a single parameter that summarizes the pollution load index and the total organic carbon distribution. The plot of this index shows that there is a small spot at 40 meters of depth in front of the former plant where the environmental stress is quite high. It is also possible to accept a separation between the external samples and the internal ones. In the external samples far from Bagnoli, the environmental stress is much lower. The spatial analysis of the ecological indexes provide important information about the variation of the ecological quality status in the area. In fact, all the ecological indexes considered have a similar trend, characterizing the marine area in front of the former plant as influenced by a bad ecological quality status, which tend to improve getting far from the contamination source. In conclusion, the internal and external stations are clearly separated based on contamination level. There is a high anthropogenic contamination in the sea sediment in front of the former plant, a strongly negative correlation between contamination and foraminiferal ecological indexes for both molecular and morphological datasets. The molecular dataset outperforms the morphological one, and lastly, the eDNA metabarcoding has confirmed to be an excellent tool in environmental assessments. Thank you for your attention.